pharmaceuticals, and biological using genetically modified microbes, fungi, plants, and animals. The application of technology include therapeutics, diagnostic, genetically modified crops for agriculture, processed food, bioremedical waste treatment, and energy production. Three critical research areas of biotechnology are providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme, critical optimal condition to engineering for a catalyst to work, downstreaming processing technology to purify the protein organic compound. Let us now learn how human beings have used biotechnology to improve the quality of human life, especially in the field of food production and health. Biotechnology application in agriculture. Let us take a look at three options that can be thought for increasing food production: agrochemical based agriculture, organic agriculture, genetically engineered crop based agriculture. The Green Revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Increasing yields have partly been due to the use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemicals, fertilizers, and pesticides. However, for farmers in the developing world, agro are often too expensive. Further increases in yield with existing varieties are not possible using conventional breeding. Use of genetically modified crop is a possible solution. Plant, bacteria, fungi, animal whose genetic genes have been altered by manipulation are called genetically modified crops. GM plants have been useful in many ways. Genetic modification has made crop more tolerant to a biotech stressor called drought, salt, heat, reduced reliances on chemical pesticides. Pest-resistant crop help to reduce post-harvest losses, increase efficiency of mineral uses by plant. This prevents early exhaustion of fertility, enhance nutritional value of food, golden rice, vitamin A enriched rice. In addition to this usage, GM has been now been used to create tailor-made plans to supply alternative resources to industry in the form of starches, fuels, and pharmaceuticals. Some of the application of biotechnology and agriculture that you will study in detail are the production of pest-resistant plants, which would could decrease the amount of pesticide used. Bt toxin is produced by a bacterium called Bacillus thuringiensis. Bt for short. Bt toxin gene has been cloned from the bacteria and been expressed in plants to provide resistance to insect without the need for the insecticide. In effect, created a biopesticide. Example are Bt cotton, Bt corn, rice, tomato, potato, and soybean. Bt cotton. Some strains of Bacillus thuringiensis produce protein that kill certain insects such as lepidopterans, tobacco, bud, worm, armyworm, coleopterans, beetles, and dipterans, flies, mosquitoes. Bt thuringiensis form protein crystal during a particular phase of their growth. This crystal contain a toxic insecticidal protein. Bt toxin protein exists the inactive protoxin, but once an insect ingests the inactive toxin, it is converted into an active form of toxin due to the alkaline pH of the gut, which solubilizes the crystals. The activate toxin binds to the surface of mid-gut epithelial cells and create pores that cause cell swelling and lysis and eventually cause death of the insect. Specific Bt toxin genes were isolated from Bacillus thuringiensis and incorporated into the several crop plants such as cotton. The choice of gene depends upon the crop and the targeted pest, as most Bt toxins are insect group specific. The toxin is coded by a gene Cry1AC named Cry. There are a number of them. For example, the protein encoded by genes Cry1AC and Cry-AB control the cotton bollworm, that of Cry1AB control corn borer. Pest resistant plants. Several nematode parasites a wide variety of plants and animals, including human beings. A nematode Merodoni incognitia infect the roots of tobacco plants and cause a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interface (RNAi). RNAi takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of a specific mRNA due to complementary dsRNA molecule that binds to and prevents translation of mRNA. Silencing the source of this complementary RNA could be from an infection by viruses having RNA genomes or by genetics elements, transposons that replicate via an RNA intermediate. Using agrobacterium vectors, nematode specific genes were incorporated into the host plant. The introduction of DNA was such that it produced both synthetic RNA in the host cell. The two RNA being complementary to each other formed a double standard dsRNA that initiated RNA and thus silenced the specific mRNA of the nematode. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. Biotechnical application in medicine. The recombinant DNA technology have been made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Further, the recombinant DNA therapeutics do not introduce unwanted immunological repression as is common in case of similar products isolated from non-human sources. At present, about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being marketed. Management of adult onset diabetes is possible by taking insulin at regular time interval. Now imagine a bacteria would available that could make human insulin. Suddenly the whole process becomes so simple. You can easily grow a large quantity of bacteria and make as much insulin as you need. Insulin used for diabetes were earlier extracted for pancreas of slaughtered cattle and pigs. Insulin from an animal source to cause some patients to develop allergy or other types of reaction to the foreign protein. Insulin consists of two short polypeptide chains, chain A and chain B, that are linked together by disulfide bridges. In mammals, including human, insulin is synthesized as a prohormone like a proenzyme. The prohormone also needs to be processed before it becomes a fully mature and functional hormone, which contains an extra stage called the C-peptide. This C-peptide is not present in the mature insulin and is removed during maturation into insulin. The main challenges for production of insulin using DNA rDNA techniques was getting insulin assembled into a mature form. In 1993, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of E. coli to produce insulin chain. Chain A and B were produced separately, extracted and combined by creating disulfide bonds to form human insulin. If a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Gene therapy is a collection of methods that allow correction of a gene defect that has been diagnosed in a children or embryo. Here, genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function and compensate for the non-functional gene. The first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. This enzyme is crucial for the immune system to function. The disorder is caused due to deletion of the gene for adenosine deaminase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement theory, in which function ADA is given to the patient by injection. But the problem with both of these lymphocytes from the blood of the patient are grown in a culture medium. Outside the body. A functional ACDC DNA using retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene is isolated from marrow cells producing ADA is introduced into cells at early embryonic stage, it could be a permanent cure. Molecular diagnosis. For effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Using conventional method of diagnosis, serum and urine analysis, early detection is not possible. Recombinant DNA technology, polymerase chain reaction, enzyme immunosorbent assay are some of the techniques that serve the purpose of early diagnosis. Presence of pathogen, virus, bacteria is normally suspected only when the pathogen has produced a disease symptom. By this time, the concentration of pathogen is already very high in the body. However, very low concentration of the bacterial virus at a time and the symptoms of the disease are not yet visible can be detected by amplification of their nucleic acid by PCR. PCR is now routinely used to detect HIV in suspected at patients. It is being used to detect mutation in gene in suspected cancer patients too. It is a powerful technique to identify many other genetic disorders. A single standard DNA RNA tagged with a radioactive molecule probe is allowed to hybridize to each complementary DNA in a clone of cells, followed by detection using autoradiobiography. The clone having the mutated gene will not appear on the photography film because the probe will not have complementary with the mutated gene. ELISA is based on the principle of antigen antibody interaction. Infection by pathogen can be detected by presence of antigen, protein, glycoprotein, or by detecting antibodies synthesized against the pathogen. Animals that have their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. Transgenic rats, rabbits, pigs, sheep, cows, and fish has been produced. Although over 95% of all existing transgenic animals are mice. Normal physiology and social development. Transgenic animal can be regulated and how they affect the normal function of the body and its development. Study of complex factors involved in growth, such as insulin-like growth factor, by introducing genes from other species that alter the formation of this factor and studying the biological effect that results. Information is obtained about the biological role of factor in the body. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. They are specifically made to serve as model for human diseases so that investigation of new treatment for disease is made possible. Today, transgenic model exists for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. Biological products. Medicine required to treat certain human diseases can contain biological products, but such products are often expensive to use. Transgenic animals that produce useful biological products can be created by introduction of the portion of DNA or gene which code for a particular product, such as human protein alpha 1 antitrypsin used to treat emphysema. Similar patterns are being made for treatment of phenylketonuria and cystic fibrosis. In 1997, the first transgenic cow, Rosie, produced human enriched protein milk 2.4 gram per liter. The milk contained the human alpha lactalbumin and was nutritionally a more balanced product for human babies than natural cow milk. Vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccine before they are used on humans. Transgenic mice are being used to test the safety of the polio vaccine. If successful and thought to be reliable, they would replicate the use of monkeys to test the safety of batches of the vaccine. Chemical safety testing. This is known as toxicity safety testing. This procedure is same as that used for testing toxicity of drugs. Transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to toxic substances and the effects study. Toxicity testing in animals would allow us to obtain results in less time. The manipulation of living organisms by the human race cannot go on any further without regulation. Some ethical standards are required to evaluate the morality of all human activities that might help or harm living organism. Going beyond the mortality of such issues, the biological significance of such things is also what a genetic modification of organism can have been unpractical results when such organisms are introduced into the ecosystem. Therefore, Indian government has set up organizations such as GEAC, Genetic Engineer Approval Committee, which will make decisions regarding the validity of GM research and the safety of introducing GM organisms for public services. The modification uses of living organisms for public services as food and medicine sources, for example, also create a problem with patents granted for the same. There is growing public anger that certain companies are being granted patents for products that make use of the genetic material, plants, and other biological resources that have long been identified, developed, and used by farmer and indigenous people of a specific region or country. Rice is an important food in the presence of which goes back thousands of years in Asia's agriculture history. There are an estimated 2 lakh variety of rice in India alone. The diversity of rice in India is one of the richest in the world. Basmati rice is for unique aroma and flavor, and 27 documented varieties of basmati are grown in India. There is reference to basmati in national text, folklore, and poetry as it has been grown for centuries. In 1997, an American company got patent right on basmati rice through the U.S. Patent and Trademark Office. This allowed the company to sell a new variety of basmati in the U.S. and abroad. The new variety of basmati had actually been derived from India's farmer varieties. Indian basmati was crossed with the semi-dwarf varieties acclaimed as an innovation or a novelty. The patent extended to functional equivalence, implying that other people selling basmati rice could be restricted by the patent. Several attempts have also been made to to patent register products and processes based on the Indian traditional herbal medicine turmeric. If we are not vigilant and we not immediately counter this patent application, other countries individuals make in cash on our rich legacy and we may not be able to do anything about it. Bioparasite is a term used to refer to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and principal concerned without compensatory payment. Most of the industrialized nations are rich financial but poor in biodiversity and traditional knowledge. In contrast, the developing and the underdeveloped world is rich in biodiversity and traditional bioresources can be exploited to develop modern application and can be also be used to save time, effort, and expenditure during their commercialization. There has been current realization of the unjust inadequate compensation and benefit sharing between developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresource and traditional knowledge. The Indian Parliament has recently cleared the second amendment of the Indian Patent Bill that takes such issues into consideration, including patent terms, emergency provision, and research and development initiative.